Hello, and welcome back to the Validity Podcast. I'm your host, Valentina Rodriguez, and this is episode 3 of season 2 because this is my second year at USF as a master's student in the exercise science program with a concentration in physique enhancement. So this is my last semester. I'm graduating in December, and we're about 3-4 weeks into the semester. And last week we talked about the molecular response to resistance training. And this week, pretty short episode because a lot of my last lecture in my neuromuscular exercise physiology course, where most of the episodes are coming from, last lecture, a lot of it was just normal review about skeletal muscle physiology and the neuromuscular activation of skeletal muscle, and we've kind of gone through that. So most of the lecture that I sat through, I won't be recapping for you guys because I've done episodes on this stuff before, like Titan and all of that. However, we did talk about a couple of regulatory genes and regulatory pathways that are relevant in the pathways that we discussed last episode. So this episode is going to build on to last week's episode. So that's a little bit about what you can expect from today's episode. In terms of personal updates, nothing crazy, still recruiting for the world's first reverse diet study. If you're interested in participating in that, then go ahead and shoot an email to reversedietstudy@gmail.com. I'm still reversing. I am just over 7 weeks post-show, ranging between 3 to 4 pounds above stage weight. Food is up, cardio's down. I'm in a good place. I'm still applying for PhD programs. I am just like in the thick of the semester, the grind. I finally filmed my first reverse dieting series YouTube video yesterday, so that should be up within the week. I made a little like protein pumpkin banana bread with some of the new P Science pumpkin products, like the pumpkin pancake mix and the pumpkin protein powder. And it smells divine. I'm taste testing it today for the video. So if it's good, we'll include it in there and I'll include the recipe. If not, then I got to keep playing around with the recipe until it's good. But yeah, otherwise, I think those are all my personal updates, super quick, and we're going to get straight into the science. So last week we discussed the signaling pathway that occurs whenever you resistance train and upregulate mTOR. mTOR then goes and via 2 parallel pathways kind of upregulates the activation or inactivation of necessary transcription factors and translation initiation factors to then increase your rates of muscle protein synthesis. If none of this makes sense, then please go back and listen to the molecular response to resistance training episode because that is necessary for all of this to be contextualized. So we know that you train, you upregulate your mTOR, and that in turn upregulates your muscle protein synthesis via the pathway that we previously discussed. However, there are some regulatory proteins and regulatory genes in place that kind of act as what we call molecular brakes in this pathway. So there was a study, an acute intervention study, that determined that there are some genes regulating translation initiation signaling and progressive skeletal muscle hypertrophic responses to resistance training. So meaning there are some genes that encode for proteins that are involved with regulating this entire pathway. So it's not just you resistance train, you upregulate mTOR, and then you upregulate muscle protein synthesis down the line. You cannot just indefinitely upregulate mTOR, because if you could, that would mean that you could train all day, every day, keep increasing mTOR, keep increasing muscle protein synthesis, and just indefinitely grow. We know that people have what we call like a cap to their natural potential. You hear a lot of people being like, oh, I won't touch PEDs until I've kind of maxed out my natural potential, my genetic potential. We don't know if that's necessary. I mean, it is a thing. We know that you cannot just indefinitely grow forever and ever and ever copious amounts. We don't know what that cap is. That cap is likely different for everybody, but a lot of what we think are the players at play here when it comes to regulating these pathways are certain, like I said, molecular brakes. So another thing that we kind of have to just kind of caveat is that we talk about mTOR being upregulated in response to lifting, and then that in turn upregulating your muscle protein synthesis via the upregulation of translation initiation. However, changes in muscle protein synthesis, or in MPS, are not always predictive of hypertrophy. We have an anabolic response to alternative stimuli such as insulin, amino acids, and those don't always relate in a linear manner to the activation of mTOR-related molecules or pathways or muscle protein synthesis. The hypertrophy of existing muscle fibers actually appears to be limited by further incorporation of nuclei from satellite cells. So this is factors beyond just upregulating muscle protein synthesis. We talked about last time how in the short term, you upregulate the efficiency of translation But in the long term, you need to increase your translational capacity. And a lot of that comes from increasing your amount of template DNA via increasing your number of nuclei via the donation of nuclei from surrounding satellite cells in order to increase the amount of rRNA so that you can increase your amount of ribosomes that you're creating. And that's your translational machinery. I hope this makes sense. Oh my God. Okay. So there's a study called Molecular Breaks Regulating mTORC1 Activation in Skeletal Muscle Following Synergist Ablation. We talked about synergistic ablation. This is essentially something done in animal models where you ablate or you remove a primary muscle, a synergistic muscle, and by making the surrounding muscles or the accessory muscles have to take on the load. So take a little mouse, a little rat, you cut out a part of its quad, and essentially the other quad muscles now have to compensate and take on the entire load of that animal walking around, living, breathing, support the body weight, whatever. And therefore, those surrounding muscles grow really, really rapidly. And so in this paper, the authors were able to identify at least 3 mechanisms that were working against the growth that was seen by the end of the period. So there were some takeaways. One, yes, synergistic ablation induced significant growth and rapid growth. And a lot of times when pathways or systems in the body go into hyperdrive or they occur more rapidly than physiologically expected, that's where you would see the turning on of certain brakes, of certain inhibitors, of certain regulatory proteins. You would see those turn on at a more significant level. And so that's why synergistic ablation was a really good model to try to identify these molecular breaks, because you're seeing rapid growth that occurs way faster than it would under normal physiological conditions. So the synergistic ablation did induce this really rapid, significant growth. And as a result of having this rapid, significant growth, at least 3 different mechanisms of regulation were identified that were working against the growth, that were regulating that growth, that were limiting that growth by the end of the study's time period. So they were able to get that rapid growth, and then proteins were turned on to stop the rapid growth or prevent it from just keeping on going indefinitely. And one of those 3 regulatory mechanisms, which is the one we're going to discuss today, is myostatin. So if you've ever heard of myostatin, or you've ever seen those like dogs or horses or cows that have genetic mutations that disrupts their myostatin, they are huge and they are jacked because myostatin is an inhibitor of the mTOR pathway. It regulates the mTOR pathway. So if you have a genetic mutation that disrupts your myostatin, that removes it, that knocks it out, that decreases its expression, whatever it may be, if it's a mutation that in some way, shape, or form limits, decreases, or downregulates myostatin, the mTOR pathway operates and occurs and runs at a greater level than would be physiologically normal, and you get greater growth than you would normally physiologically expect. We looked into a case study that was published in 2004, and it's called Myostatin Mutation Associated with Gross Muscle Hypertrophy in a Child. Okay, so essentially this was a case study done on a child that was born to an athletic woman whose father, well, the child's father was unidentified, but basically at I want to say 6 days old, they were able to identify like overly developed quad muscles, calf muscles. So this newborn had skeletal muscle characteristics that are not expected for a newborn. You do not expect a newborn to have developed quads that protrude from their legs or calves or anything of the sort. And it turns out that this child had a mutation in their GDF8 gene, which is the myostatin gene. And this gene is expressed almost exclusively in muscle cells, mostly skeletal muscle cells, but we do also see it in cardiac muscle. And so getting rid of this myostatin gene or having a mutation in this GDF8 or myostatin gene results in almost a doubling of your muscle mass and a doubling of your physical, visible size and shape. You don't just look bigger, but you have the increased muscle mass to support the bigger size that you now have. This mutation is very, very rare in humans. This is why there was a case study done on this child. We tried to find a follow-up because this was published in 2004 and we could not find one. But they did follow him up until, I want to say, 4.5 years old. So from 6 days old to 4.5 years old, they followed him. He obviously had this loss of function mutation in the myostatin gene, and they were specifically closely monitoring his heart for cardiomyopathy or cardiomegaly because since you do express myostatin in your cardiac muscle too, they were kind of concerned that they might have this enlarging of his heart that would give him heart problems. But no health problems were seen up until 4.5 years old. Again, we don't really know where he is now or how he's doing, but for the first 4.5 years of his life, this loss of function mutation in myostatin did not elucidate any other health concerns. He was just jacked and healthy, which is kind of crazy. And this case study, we had seen this kind of in animals, this loss of function mutation result in animals and mice and cows and dogs and horses be really jacked, essentially. And this was kind of the first time that this mutation was identified in a child. And it provided strong evidence that myostatin does play a role in regulating muscle growth in humans because the loss of function of this specific protein resulted in an increase in muscle mass. So what happens when you overexpress myostatin? So the opposite, this baby, this child had a malfunctioning myostatin gene. What if you have a myostatin gene that is overly expressed, that is present in abundance? Then you get muscle wasting and muscle loss and atrophy, and that is also very dangerous, but we don't have a case study on anybody who has that I'm aware of. So obviously, given this baby, given what we've seen with animals, myostatin is a really enticing molecule of interest for pharmacological intervention for diseases that cause muscle dystrophy, muscle wasting, muscle atrophy, etc. So if you can find a way to inhibit myostatin in humans, theoretically, you can increase their mTOR signaling pathways. You can upregulate those and increase the muscle mass, the hypertrophy of whoever needs it. This would be specifically used in disease states where you get muscle dystrophy and muscle wasting. However, we don't have a lot of promising evidence whenever interventions have tried to be created or tested for myostatin specifically. There is a researcher, her name is Catherine Wagner, and she seems to do a lot of work in the myostatin space, and she has a paper published in 2005 called Muscle Regeneration Through Myostatin Inhibition. And essentially, she proposed that the best way to inhibit myostatin is by creating neutralizing antibodies that inhibit the myostatin signaling. So not really targeting it at the genetic level, like letting the gene be expressed, but then blocking the pathway that protein would then act on. But the research is still ongoing and it hasn't yielded anything meaningful. She also created a review of all the research out there on myostatin inhibitors and myostatin inhibition. And it's actually titled The Elusive Promise of Myostatin Inhibition for Muscular Dystrophy, because so far, again, nothing meaningful has really come out. Myostatin inhibition ameliorates phenotypes of many, but not all, mouse models with muscular dystrophy. So in animal models that they've tested this, not all of them had that dystrophy ameliorated, which means the condition was improved, but not all of them did have that improvement. Clinical trials in humans have not been efficacious, and most preclinical trials, which would mean the animal model, so you go from animal model, you get enough evidence, then you apply for a bunch of things with the government until they grant you the ability to run clinical trials. There's a lot of steps before you can get this to humans. And preclinical trials in animals is more promising than anything we've seen in clinical trials in humans. So it just seems that there's a lack of translation between preclinical trials and clinical trial results that just suggests that maybe the mouse is a poor model for muscular dystrophy, because clearly what we're seeing in mice is not translating to humans. So maybe we need a new animal model that can give us a different just way of doing things at the preclinical level that will better translate to human trials. And this author also concluded that myostatin inhibition as a monotherapy for muscular dystrophy related diseases is improbable, but maybe in combination with other therapies, with other, molecular regulators that they find later in science, whatever, it could be a more promising intervention. So that's kind of myostatin, and that's one of the regulators of the molecular pathway that we discussed. The other regulatory gene that we discussed comes from a paper that I'm going to link below, and essentially it's looking at microRNA expression and trying to see how microRNA expression differed between people who were either high responders to resistance training or low responders to resistance training. High responders being people who very obviously have a greater hypertrophic response to training than others. And if you're thinking about this just in the general sense, you might think, okay, I have a friend who just started lifting and they're really jacked, like they are just genetically blessed, and I've been lifting for 8 years and it doesn't even look like I lift. That has nothing to do with your work ethic, has nothing to, it could have to do with your training and all of that, but most likely it's just down to the genetic differences. And one of the ways in which those genetic differences are manifested is through the differential expression of different microRNAs. You don't need to know too much about what a microRNA is. Just know that microRNAs are essentially regulators of the way that genes eventually get turned into protein. So if you regulate that entire pathway in any place in the pathway, you change the number of protein that's produced. So they found that low responders to resistance training, so people who don't grow too much when they train, had down-regulated levels of microRNA 378, 29A, and 26A. These were all down-regulated in low responders, but were unchanged in high responders to resistance training. An additional analysis suggested that the loss of expression of microRNAs 378, 29A, and 26A, you do not need to know those numbers at all, just these are the specific ones they found in the study, appears to be compensatory, meaning that these microRNAs are regulators of protein expression, and they target the muscle hypertrophic pathways. And we don't know too much beyond that about what they do, but we do know that in these low responders, they had downregulated levels of these 3 key microRNAs that seem to play a role in the muscle hypertrophic pathway, whereas in high responders, the levels of these microRNAs were unchanged. And this is, again, just one of the ways in which you cannot determine your level of microRNA 378, 29A, or 26A expression. Like, you're just born with a genotype that either codes for high expression of these microRNAs or low expression of these microRNAs. And so when we're thinking about just general genetic differences between people, we talked a little bit about how genetic differences can influence a phenotype. A phenotype is your physical manifestation of your genetic basis genetic code. So if you have one genotype, it will result in a specific phenotype versus if you have the other, etc. And so to recap, we identified 2 potential molecular regulators of this muscle hypertrophic pathway that's mediated through mTOR. These regulators, one of them being myostatin and the other one being these identified 3 microRNAs. And we can appreciate that the way that we express these different molecules, so how much myostatin you express and especially how much of these microRNAs you express, is determined by your genetic code. It's genetically predetermined. And these genetic differences are what eventually result in some people being naturally really jacked, some people being naturally really small, some people responding really well to training, some people not. And we have to keep in mind that we know that we're also different and we all have different genetics and this and that and whatever. But I think it's really helpful when we can kind of break it down to like, okay, we're different genetically, and you can kind of say we might literally have differences in our microRNA 378, 29A, or 26A levels, and that's why I am or I'm not responding as well as you to resistance training or this or that. Like for me, it helps a lot to be able to contextualize all these like things and concepts and philosophies that we throw around in the world that we live in of fitness and health and social media and bodybuilding and lifestyle and health, whatever, lifting. You get it. It helps me to be able to like kind of bring it down and physically contextualize it into a pathway, into a molecule, into a regulator, into a gene, etc., whatever it may be. So yeah, so today's episode, I know, really short, but just wanted to kind of chit chat through 2 potential regulators of the pathways that we discussed last episode. And the next episode, which I'm literally about to record because I'll be out of town, so I want to get a head start, is going to be on just general genetic differences and how those play a role in our skeletal muscle phenotypes and just in our general manifestations of strength, of hypertrophy, of size, of exercise performance, etc. Should also be another short episode, but with that, I hope all of this made sense. I hope you guys have an amazing week. If you have any questions, as always, email my podcast email or DM my podcast Instagram. Everything is below. My one-on-one coaching application link is below. The reverse diet study email is below. It's reversedietstudy@gmail.com. And until then, I hope you guys enjoyed this, and I will see you in the next episode.